So first I would like to thank the organizers for putting together such a lovely meeting. It was nice to see again so many people. So I'm going to talk about the interaction of Botrytis cinerea with grapes from both susceptible and tolerant cultivars. So actually, this project started some years ago. We performed the infections of trincadeira berries. It's a Portuguese cultivar, widely used in Portuguese wines, but extremely susceptible to Botrytis cinerea infection. It has this very much compact cluster. And of course, we had heavy symptoms of infection. We performed these infections in an experimental vineyard located in Lisbon. But at first, we were very surprised that we could see heavy symptoms of infection in green berries, which was confirmed by a strong reprogramming of both the transcriptome and the metabolome. And then we started to wonder what would be with other cultivars that are tolerant, how the reprogramming of the metabolism would occur in these cultivars. And also, we were very much interested with the RNA sequencing to see how the metabolism of the Botrytis would also behave. So, we performed these infections at EL29, which corresponds to the pea-size berries, and we collected at several developmental stages, but the main target was the green berries at AL32. What we could see is that, of course, in the tolerant cultivar, again, we had heavy symptoms of infection in the green berries, but with Syrah, we could only notice heavy symptoms of infection when they were already ripe. So we have collected these samples at green, veraison, and harvest stages, and we used it for targeted hormonal profiling, transcriptomics using RNA sequencing, and metabolomics using two platforms for GCMS. And here you can see clearly that at harvest stage, the symptoms of infection were huge in trincadeira, but Syrah already exhibit from the visual inspection some symptoms of infection as well. And we confirmed this with, by qPCR using the genomic DNA of the fungus. So, regarding the hormonal profiling, we have evaluated the content of several hormones. The salicylic acid actually stand out because it was not classical associated with a response to necrotrophic pathogens. However, and I will highlight mostly the results obtained for the green grapes at EL32, you can see, you could see that at EL32, there was a higher basal content of salicylic acid. It was even activated at veraison, but when they were exhibiting both symptoms, the salicylic acid content was quite similar. Moving forward, regarding jasmonates, while for the precursor of jasmonic acid, or PDA, we could not see differences in basal content. It was increased in trincadeira, but most importantly, the conjugate with isoleucine, which is described as the most important in the signaling properties associated with defense response. It was also higher the content in the tolerant cultivar. We did some integration also with some qPCR, targeted qPCR. This was quite interesting because this transcription factor has been associated with the response to Botrytis cinerea, and mutants of these MYC2 have been reported to present reduced resistance to Botrytis cinerea. So we have two more classes of hormones, auxins, and here again I highlight the higher content, basal content of auxins, which have been already for some years as emerging as also main regulators of defensive response, also in interaction with other growth regulators such as osmotic acid. And we could also see that genes coding for enzymes involved in its metabolism were also with a trend to be more expressed in the tolerant cultivar. Here was more in the infected. So it was clear that the metabolism was different between these two cultivars, the grapes of these cultivars, and it seems that salicylic acid, the jasmonates, and auxins may be involved in defensive response. For abscisic acid, we had indications that was probably more involved in susceptibility and not so much in resilience. It was higher the content here, but we believe, given its role in promoting ripening, which increases very much at veraison, we predicted that it was probably involved in promoting susceptibility by accelerating fruit ripening. So coming to wrap up this part, we could see that there was a different metabolism in between the cultivars that was putatively involved in the capability of defensive response. And I will just go through this putative model very fastly. Basically, you have basal active defense being activated prior to infection. In here, it's mostly down-regulated, and then everything changes when the berries are infected for trincadata. It keeps pretty much the same in cinerea. So, given these results, we were starting to wonder, okay, we had a different hormonal metabolism, but what about other defensive metabolites? So we did GCMS. And we could, here by this time we were focusing on the green grapes. And of course we could see a lot of different content in several compounds, phenylpropanoids, fatty acids, a lot of fatty acids. This is quite interesting because it depends on the pathogen. We compare it with previous experiments that we did with other pathogens. But what I would like to highlight is that for cinerea, there is absolutely no changes, and for trincadata, they were activated under infection. So here in this slide, we present also some putative markers of infection, and it's clear that cinerea doesn't change. In some cases, the same compound that was in higher content in cinerea increases in trincadata after infection, and we highlight these ones, in particular certain fatty acids, alpha-tocopherol, and catechins that were actually higher content in cinerea and may play a role in the defensive process. Moving forward for the RNA sequencing data, again the same thing. I will not go into detail into these functional classes, but basically we could see a strong reprogramming of trincadata under infection, and we could not see so much in cinerea under infection. And we had, of course, the genes being activated involving cell wall metabolism, transcription factors, phenylpropanoid metabolism, so a bit of classic results. What was interesting in this project was, in fact, the metabolism of the Botrytis cinerea. I'm not sure if we were really surprised, but anyhow, what we could see, there was active transcription in cinerea, at this early stage of infection, where we don't see visible symptoms yet. And we had a lot of genes being expressed involved in cell wall metabolism, the classical enzymes, active transcription, autophagy, oxidative stress. So genes involved in virulence were highly expressed in sera and not so much in Trincadera. With Trincadera were more genes related with advanced stage of infection and host colonization. So, to finish, we can say that in the plant we have these basal resistance based on resistance proteins, phenylpropanoid metabolism, hormonal signaling, and different hormonal content, as I already showed you. So this in the plant. And then when you move to the fungus, we could see that indeed we confirm previous results. It is clear that the berries are pretty much infected in Trincadera, but what was interesting is that we had a highly virulent fungus present in sera. So actually it makes sense that the fungus, once you manage to get in, it gets more relaxed and may lose virulence-associated mechanism when the environment is actually prone to successful infection. So in our model, what we could see is that in Botrytis we had 3 more times genes expressed in the planta, comparing with Trincadera, but look here, the difference is enormous regarding the transcription reprogramming in the susceptible, almost nothing, comparing with the Trincadera post-infection. Here we could see, as I already described you, the basal immunity related genes, and also, of course, the different content in hormones and metabolites. And here it was activated after infection. Here we could see that the Botrytis cinerea was highly virulent, as I previously said, exhibiting a lot of genes already described as important in virulence. And here, since the pathogen already managed to get in, and we could also see differences in genes coding for cell wall metabolism, we could see that it was a much less virulence-associated metabolism in this cultivar. Of course, we want to confirm this in other susceptible and tolerant cultivars, but anyhow, it makes sense that if the pathogen needs to get in, that it activates more virulence-associated mechanisms. So finally, I would like to introduce you to these concluding remarks. So pre-activated defenses in sera are likely to be responsible for its resilience against Botrytis cinerea attack. That involves the sexual defense in sera, a wide activation of signaling pathways and carbohydrate metabolism in Botrytis cinerea. That it also put in evidence the different virulence-associated metabolism regarding fungal growth, signaling, as I already mentioned, which may be important for early stages of infection and may eventually be important as targets to figure out the mechanisms of successful infection of these pathogen that infects such a wide range of plant species. And of course, this would be could be used for functional characterization in the pathogen. So finally, I would like to thank the people involved in this work from the science faculty. We also had an important collaboration with a phytopathology group, also an institution in Lisbon. And of course, the precious collaboration of the Max Planck, both from Joachim Kopka and Axel Mithofer, for the metabolomics and the hormonal profiling. Thank you for your attention.